breakdown of glycogen, the major carbohydrate storage compound in humans. We'll also look at the pentose pathway, which provides the mechanism for generating NADPH. The glucose polymer glycogen serves as the major form of carbohydrate storage in the liver and muscle of humans. Glycogen has a backbone of alpha-1,4-linked glucose units, with alpha-1,6 branches occurring about every 10 to 12 glucose units. The largest stores of glycogen are found in liver and muscle. The glycogen polymer contains about 90% alpha-1,4-linked glucose units and 10% alpha-1,6-linked glucose units. The extensive branching of glycogen provides many non-reducing ends which are subject to enzymatic hydrolysis reactions. There's only one reducing end, where a glucose unit could undergo mutarotation to the open-chain form. The enzyme phosphorylase A attacks the non-reducing ends of glycogen, releasing alpha-1,6-linked glucose units as glucose 1-phosphate. This reaction proceeds in the direction of product formation. Phosphorylase A cannot attack glucose units that are near to alpha-1,6 branch points, so a transferase enzyme and an alpha-1,6 glycosidase enzyme converts the 1,6 branches into additional linear 1,4-linked glucose units. That transformation allows phosphorylase A action to continue further into the glycogen molecule. The glucose 1-phosphate units produced by phosphorylase A are converted to glucose 6-phosphate by the enzyme phosphoglucomutase. The glucose 6-phosphate can then enter into the glycolysis pathway. Glycogen can be synthesized from glucose using different anabolic enzymes from those used for glycogen breakdown. The synthesis of glycogen is the reversal of glycogen breakdown. The cleavage of glycogen is an energy-releasing reaction, so energy input is necessary to make the reverse reaction proceed. Glycogen synthesis employs a set of enzymes to reverse the action of phosphorylase A. Glycogen synthesis proceeds through an activated UDP glucose sugar nucleotide intermediate. The first step in glycogen synthesis is catalyzed by UDP glucose pyrophosphorylase. In that reaction, glucose 1-phosphate reacts with UTP to form UDP glucose and release an inorganic pyrophosphate. The pyrophosphate released in the previous reaction can be hydrolyzed to form two inorganic phosphates. The hydrolysis of pyrophosphate is an energy-releasing process which removes the product of the previous reaction and helps to drive the overall reaction sequence towards product formation. In the second step in glycogen synthesis, a synthase enzyme transfers a glucose unit from UDP glucose to the non-reducing end of glycogen. UDP is released as the other product. From this overview slide, it's clear that glycogen breakdown and synthesis involve different enzyme systems. There are a number of important reasons why this is necessary. The use of different enzymes allows both breakdown and synthesis of glycogen to be energy-releasing processes. This is necessary so that both reaction sequences can proceed in the direction of product formation. The use of alternate enzyme systems allows both systems to be subject to hormonal regulation. The net result of hormonal regulation is that both systems do not function simultaneously. If they did, the net result would be a futile cycle of enzyme activity which would waste energy and yield no net product. One cycle around the system results in the net hydrolysis of one ATP equivalent. Hormones are organic compounds that are synthesized in one tissue and are transported through the blood to a remote site of action at another tissue where they exert an effect on metabolic processes. Adrenaline, also called epinephrine, is an example of a low molecular weight hormone that's derived from an amino acid, specifically the amino acid tyrosine. Epinephrine stimulates a series of reactions that result in the activation of the catabolic enzyme phosphorylase A, which degrades glycogen. The regulatory cascade includes a number of features which you'll encounter often in biochemistry. These features include the binding of a hormone to activate an extracellular receptor protein, the activation of a G protein by interaction with the activated extracellular receptor, the activation of adenylate cyclase by interaction with the G protein, the conversion of ATP to cyclic AMP by adenylate cyclase enzyme, the activation of a protein kinase enzyme by cyclic AMP, the activation of phosphorylase kinase by a phosphorylation reaction catalyzed by the cyclic AMP sensitive kinase, the conversion of phosphorylase B to phosphorylase A by phosphorylation by the phosphorylase kinase, the cleavage of glycogen by phosphorylase A. Epinephrine also stimulates an enzyme cascade that results in the inactivation of the anabolic enzyme, glycogen synthase, which adds glucose units to glycogen. This regulatory cascade includes most of the same features seen in the activation of glycogen catabolism, except that the end of this cascade results in the inactivation of an enzyme and the suppression of glycogen synthesis. The dual function of the epinephrine cascade in activating glycogen breakdown and simultaneously inactivating glycogen synthesis prevents these reactions from catalyzing a futile cycle in which energy is wasted. Adenylate cyclase converts ATP to the second messenger, cyclic AMP. Second messengers are intracellular signaling agents that are often synthesized or degraded in response to the action of an extracellular hormone. In the cell, the adenylate cyclase reaction is essentially irreversible because one of the products, inorganic pyrophosphate, is rapidly degraded by a pyrophosphatase enzyme. This strategy, the breakdown of inorganic pyrophosphate, is used often in biochemical pathways to force a reaction to go in the direction of product formation. The pentose phosphate pathway is an alternate pathway to glycolysis that provides a source of NADPH for use in reductive biosynthesis, and it also generates pentose sugars. Some of the reactions of the pentose pathway are integrated with glycolysis and gluconeogenesis. All the reactions of the pentose phosphate pathway take place in the cytosol. Most anabolic biosynthetic pathways are also reductive. That is, the products of biosynthesis are generally more reduced, having a higher percentage of hydrogen atoms than the starting materials. The reducing power for anabolic pathways comes from NADPH. Fatty acid biosynthesis and nucleotide biosynthesis are examples of anabolic pathways that utilize NADPH. This graphic shows one way of representing the pentose phosphate pathway. This is a simplified version of mode 4 of the pathway with a number of the reactions omitted. Mode 4 of the pathway is operative when the cell needs NADPH and ATP, but does not need ribose 5-phosphate for nucleic acid biosynthesis. We can break this form of the pathway down into two phases. The first phase of the pathway, which runs from glucose 6-phosphate to ribose 5-phosphate, involves the oxidative generation of NADPH. The second phase of the pathway involves the non-oxidative conversion of 5-carbon sugars to intermediates of glycolysis, such as fructose 6-phosphate, glyceraldehyde 3-phosphate, and pyruvate. In the oxidative phase, the starting material, glucose 6-phosphate, is oxidized to ribulose 5-phosphate, and in two of the reactions, the cofactor NADP+ is reduced to NADPH. In fact, all of the NADPH produced by the pentose phosphate pathway is synthesized in the oxidative phase. The net reaction of the oxidative phase is glucose 6-phosphate plus 2 NADP plus water yield ribulose 5-phosphate plus 2 NADPH plus 2 protons plus CO2. The pathway could stop right here if it were not for the fact that the cell only needs a limited supply of ribulose 5-phosphate, probably much less than is produced to meet the need for NADPH. So further reactions are needed to convert excess ribulose 5-phosphate into usable intermediates such as pyruvate, which ultimately are oxidized to carbon dioxide. In other modes of the pentose pathway, not shown, excess ribulose 5-phosphate is converted back to glucose 6-phosphate by gluconeogenesis. In the non-oxidative phase, the 5-carbon sugars xylulose 5-phosphate and ribose 5-phosphate are converted to fructose 6-phosphate and glyceraldehyde 3-phosphate. This phase of the pathway involves a convoluted anestomosizing series of reactions which nature designed to confound the naive. The products of this phase are mainstream metabolites that can enter glycolysis or gluconeogenesis. The first reaction of the oxidative phase is catalyzed by glucose 6-phosphate dehydrogenase. In this reaction, glucose 6-phosphate in the hemiacetyl form is oxidized to 6-phosphoglucanodelta-lactone, a cyclic ester, coupled to the reduction of NADP+ to NADPH. It may be a little hard to visualize because both the substrate and the product are in the cyclic form, but the reaction is the equivalent of oxidizing an aldehyde to an acid. The second reaction in the pathway, catalyzed by lactonase, is a ring opening which converts the lactone to the open-chain carboxylic acid, 6-phosphogluconate. In the next reaction, the 6-phosphogluconate is decarboxylated and oxidized to ribulose 5-phosphate. The reaction is coupled to the reduction of a second molecule of NADP+ to NADPH. At this point, we can consider that the oxidative phase of the pathway is finished because the synthesis of NADPH is completed. If the synthesis of NADPH were the only consideration, the pathway could stop here. The problem is that stopping at this point would leave a big pool of ribulose 5-phosphate in the cell. This would not only waste energy, but the accumulation of so much ribulose 5-phosphate within the cell would probably be toxic. For that reason, the pathway continues into the much more complicated non-oxidative stage. The purpose of the non-oxidative phase is to convert the 5-carbon intermediate ribulose 5-phosphate into other useful intermediates that can be used in glycolysis. It takes a series of 5 convoluted reactions to accomplish that task. The ribulose 5-phosphate is first converted into 2 different 5-carbon compounds, ribose 5-phosphate and xylulose 5-phosphate, by 2 separate enzymes. These 2 5-carbon intermediates are then converted to glyceraldehyde 3-phosphate and fructose 6-phosphate by a series of 3 reactions. As a first step in the non-oxidative phase, the ketone ribulose 5-phosphate can be isomerized to the aldehyde ribose 5-phosphate. The reaction involves the formation of an unstable enediol intermediate in which 2 adjacent carbon atoms containing hydroxyl groups are connected by a double bond. The enediol decays into an aldehyde, which is stable. The isomerase reaction goes through an enediol intermediate. Similar enediol intermediates occur in 2 isomerization reactions that we've seen previously in glycolysis. The mechanism as shown here is exactly analogous to that of phosphoglucose isomerase and triose phosphate isomerase in glycolysis. Learning to recognize such similarities in mechanism will help you memorize new reactions as variations on a theme rather than completely new phenomena. The top part of the pentose pathway to this point is sometimes referred to as the oxidative phase or mode 2. We're not including the formation of xylulose 5-phosphate in this mode. The products are 2 molecules of NADPH, 1 molecule of carbon dioxide, and 1 molecule of ribose 5-phosphate. If the cell always needed both NADPH and ribose 5-phosphate in the exact ratio of 2 to 1, then this could be the end of the pathway. However, the need for these two substances in that precise ratio is not generally the case. Often the two products are needed in different ratios, or one is needed and the other is not. Thus, there's a necessity for modes of the pathway other than mode 2. That is where the non-oxidative phase of the pathway comes into play. At the beginning of the non-oxidative phase of the pentose pathway, ribulose 5-phosphate is converted into xylulose 5-phosphate by an epimerase reaction. This reaction simply changes the chirality of the hydroxyl group at the number 3 carbon, converting ribulose 5-phosphate into xylulose 5-phosphate. Now we launch into the non-oxidative phase of the pathway. Up to this point, the sequence of reactions has been relatively linear and more or less easy to follow. With the exception of the conversion of ribulose 5-phosphate to xylulose 5-phosphate, the reactions are all in an exact linear order. We will now show you a series of reactions that are individually straightforward, but when we put them all together, it creates a convoluted pattern. There are bifurcations and conjunctions designed to confuse the unwary. In the transketolase reaction, the 2-carbon fragment coated in red is transferred from xylulose 5-phosphate to ribose 5-phosphate. The products of this reaction are glyceraldehyde 3-phosphate and cetahepulose 7-phosphate. In this reaction, 2 C5 compounds are converted to 1 C3 and 1 C7 compound. The cetahepulose 7-phosphate is a new bird, and a big bird at that, with 7 carbons. Its stereochemistry is derived from the ribose 5-phosphate, with the C1 carbonyl carbon of the ribose 5-phosphate becoming a hydroxyl group on C3 of cetahepulose 7-phosphate, with an orientation to the left. The reaction is reversible, and under some modes of the pentose phosphate pathway, it will actually function in the reverse direction. This structure of thiamine pyrophosphate is here to remind you that transketolase uses this coenzyme to transfer the 2-carbon fragment from xylulose 5-phosphate to ribose 5-phosphate. The site of attachment to the coenzyme is coded in red. This is the same cofactor that was used to transfer a 2-carbon fragment in the pyruvate dehydrogenase reaction. The next reaction involves the 2 products of the previous reaction, cetahepulose 7-phosphate and glyceraldehyde 3-phosphate. The transaldolase enzyme transfers a 3-carbon fragment coated in red from cetahepulose 7-phosphate to glyceraldehyde 3-phosphate, producing erythrose 4-phosphate and fructose 6-phosphate. In this step, 1 C7 and 1 C3 compound are converted into 1 C4 and 1 C6 compound. Finally, and it can't come too soon, the erythrose 4-phosphate product of the previous step reacts with a second xylulose 5-phosphate from the phosphopentose epimerase reaction to give glyceraldehyde 3-phosphate and fructose 6-phosphate. This reaction is catalyzed by the same transketolase enzyme we talked about before, but one of the substrates, erythrose 4-phosphate, and one of the products, fructose 6-phosphate, are different. The reason for all these bizarre manipulations is to produce stuff that can be metabolized in core reaction pathways, for example glycolysis, instead of building up a product such as ribose 5-phosphate that's a useful metabolite, but only in limited quantities. The glyceraldehyde 3-phosphate and fructose 6-phosphate can be used in virtually unlimited amounts because they're intermediates of glycolysis. Chemists mirror nature in that they have a compulsion to balance reaction pathways. This diagram gives a carbon balance for the non-oxidative phase of the pathway. If you do your bookkeeping, you'll see that there are 3 5-carbon compounds going into the process for a total of 15 carbons. There is one 3-carbon product and two 6-carbon products for a total of 15 carbons. Okay, they balance. I can relax now and move on. Take one last look at the non-oxidative stage of the pentose pathway. Try to identify where and how the 3 5-carbon compounds enter into the pathway. Then identify the 3-carbon product and the two 6-carbon products. Sometimes we go to the ocean, that is the Pacific coast of Washington State, which is about 70 miles from our house. That's the real ocean coast, not the puny little 30-mile-wide Straits of Juan de Fuca, which runs by Port Angeles. On the Pacific coast, you can experience the effects of thousands of miles of open ocean water crashing against the shore. What always amazes me is that there are so few people on the beach. Thousands of people will flock to theme parks and wait for hours in line for a cheap thrill, but only a handful of hikers walk these beaches, where you can experience some of the greatest marvels of creation. Go figure. Anyway, being a constitutive misanthrope, I hope that this trend continues for my lifetime. I enjoy seeing the bonsai-like trees that grow from rocky sea stacks. There are hundreds of these rocky little appendages clinging to the Pacific coast. I love them all. This is the same beach as in the previous slide, only on a foggy day. The rising mist gives the scene a very ethereal quality. This type of rock with its pattern of erosion is fairly common on the ocean beaches. I think the holes are caused by marine organisms that colonize the rocks and secrete acid. The hole in this driftwood frames a typical scene with sea stacks and tide pools. These young women are braving some very chilly waters to take a refreshing dip in the surf. This family seems to be looking for something. Maybe it's a migrating whale. It's very typical to have intermittent sunshine and rain on the Pacific coast. That produces an inordinate number of rainbows. Logging is a major industry on the Olympic Peninsula. As a result, many of the beaches are littered with all sizes of driftwood, including many very large logs. This flock of gulls is hunkered down near the shore. There must be some source of food nearby, but what is it? On a partially cloudy day, the sunlight filtering through the clouds lends the beach an air of mystery. Because these ocean beaches front thousands of acres of rainforest, streams and rivers of all sizes empty into the ocean here. This tiny seasonal waterfall will dry up in the summer. Who made these tracks along the beach? I like this photo so much, I'm using it as my wallpaper. The coastal shallows are home to ubiquitous kelp beds. In stormy weather, the kelp strands break loose from their moorings on the ocean floor and accumulate on the shore. What better place is there for solitude and meditation than a deserted ocean beach? I take great joy in the twisted, wind-blown trees that survive on the seaside rocks. Sometimes these stunted trees grow to be hundreds of years old. Seeing the small figures in this photo gives you some perspective as to how large some of these sea stacks really are. These people are enjoying a stroll on the beach in the midst of the mist. People walk the beaches looking for rocks, driftwood, or sometimes for just a bit of exercise. We have to stop meeting like this. This family is having some fun on the foggy beach.